Hello everyone, I'm going to be going over DNA replication and DNA expression. Now this little guide will explain to you what's going on. So here we have DNA, and on the DNA we have nucleosomes. Now, what nucleosomes are exactly doing to the DNA is that they're making it too compact. And what this means is that it doesn't allow for the DNA to be translated or transcribed. Now, so how do you start DNA replication? Let's go over the general basic idea. We have DNA, and normally it's always replicated in where the S phase. There's the S phase in the cell cycle. There's the G1, the S, the G2, and the M phase. We have these proteins. One's called DPI, and DPI signals E2F, which is an enhancer by itself. If we go back to cell cycle, we know that E2F with retinoblastoma protein is not an enhancer, but it's a repressor. So E2F stimulates MCM and CDC6 to begin. Now, to begin what? But it's blocked, right? The nucleosome is blocking the DNA strand at the moment. So how do we fix this? How do we fix this? So we incorporate transcription factors, and these transcription factors, there's many types of transcription factors. Most of them are just different types of processes, such as like we go into the G protein coupled receptor complexes or the tyrosine receptor complexes that recruit like these types of transcription factors. But for now, we're going to stick to E2F. Now E2F does what? Binds the nucleosome from the DNA. Now, not completely, it doesn't completely unbind it, but it kind of moves it over. Now, what's this look like? Let's blow this up a little bit. Actually, first let me draw my E2F protein. Yeah, my E2F protein, there we go. E2F, right there, cool. Now, put this control there. So, this right here is relaxed after we bind E2F. So let's erase this. The enhancer, the E2F enhances this. Now, let's blow this up. So we got our DNA. That's a good color, right? Oh, well, yeah, that's where the nucleosome is. Let's put that there for sure. And it's basically real. Make sure that you guys understand that the nucleosome isn't gone, it's just relaxed. Now, in there we have this little region called the promoter region. Also, it's called the Tata box, is where is the promoter region. Now, this region is important because this region is where DNA polymerase 2 can bind to. This is the Tata box promoter region. It's very important. And what now? What now we're going to go over is E2F is a low TF2B D. Now, what does TF2D do? This is what calls RNA polymerase 2 to bind to the Tata box. So the RNA polymerase can't just go bind. RNA polymerase 2 can't just go bind. It needs a signal. Just like anything else, you need a signal sequence so that something else can happen. Phase that allow this. So TF2D is one of the TF2D calls RNA polymerase 2 to bind to the Tata box. Now, what's this? This is where we start our transcription process. However, many factors to this. You can also inhibit this process. How? Well, we have these inhibitory proteins, such as this one, such as E2F with segmented with bound to RB, can negatively stimulate this process and actually prevent transcription. The nucleosome, as in here, to not leave, to not open up this gate, to open up this little portion of the DNA so that proteins can bind to it. So, and right there's our RNA polymerase too. Let's put another color. This will be your enhancer region, your repressor region. Now, enhancers and repressors normally bind. So say you have in the middle the promoter region, where that's where the DNA polymerase 2, RNA polymerase 2 is. Upstream of that, you have near the 5' end, you have where the enhancers are mainly located. And then downstream is where mainly you'll see your primary transcript takeaway. But you want a little bit. And also you have your poly A signal, always at the 3' end. This allows for, you need this going from 5' to 3' basically. Let's go back to our regular mRNA strand that we had. Just 1 second. This orange color. So many colors to choose. So find these base pairs. We'll have, say, TAC, GT, CA. We'll put template strand on the top, coding strand on the bottom. And these are important. The template strand is important just because it's used for base pairing. So we need that for base pairing. That's mainly what its job is. And the coding strand, it's not used during transcription at all, but we read DNA from the coding strand. So whenever you're given that big box of DNA of different types of amino acid groups, you read this from the coding strand. So you get A, T, G, C, A, G, T. There we go. Next. Now remember how I told you the template strand is basically there for us to do what? This allows us to start our transcription of the AUG, which is the start codon. AGU. There we go. So remember AUG, start codon. Let's get our RNA polymerase 2 in there and show that we're moving towards the 5' end on the other side. And how this gets stopped is by CPSF. This CPSF cleaves polymerase 2. After you do this, another important thing that you should be aware of is base pairs after initiation. After 10 base pairs have been coded for, after of AUG, initiation stops, and then we go to elongation. Now, initiation is complete when you start elongation. So when you start elongation, you don't have initiation anymore. And then after an elongation, you go until there's a stop codon. Until you get to the stop codon, and then that's where termination happens. Now, important note, splicing as well. So, now we gotta get the translation. A few things still have to happen. And this is where I was talking about splicing. Now, splicing is very important, and the reason why it's important is because there's so many different factors that it involves. Diseases come into play here as well as other things. But to get the start of it, let's draw our RNA strand. Now 5' to 3', and this is our primary RNA strand transcript now. So GU 5' donor site, and then you got your AGA 3' acceptor site. These are important. Splicing to occur, you need these two sites. Without these two sites, you can't really splice the introns out of the RNA transcript. So first of all, they have to join. These two sites join. How do they join? They join through these things called SNRDSs, and they form a lariat. So a lariat just looks like a boomerang. I'm not going to draw it, but that's basically it. So you got your exon 1 and your exon 2. So after those two have been spliced out, which are basically the introns of it, you have your introns in the middle. They were spliced out. Remember the AG, UG, they get stick together and they leave. You have their two exons. So you don't code anything for introns, they're left. So you got your exon 1, your exon 2, you got your UTR regions, 5' cap, and your poly A tail. Let's add our PABP. It's used to, it's needed to get into the cytoplasm. It's kind of like a code, or like that thing that you need, that code that you need to get into the club, man. So you got your transcription factors, and the transcription factors also help protect the DNA, the RNA, and the transcription factors bind on the cap site, the UTR sites, and the UTR site on the 3' end. Now, the transcription factor on the 5' site is a little bit more important than the one on the 3' site, because it helps circularize the RNA, and this circularization is going to be important for when we get into translation. We go to translation, and translation is where we get our peptide sequence. Now, the reason why I'm going over this I go over this a little bit more in detail, because a lot of books and other videos, they like to go over it, but they don't go over it with the specific enzymes that you need to know. Now, this is where the enzymes come into play. So if you knew everything beforehand about transcription, you could just skip to this part of the video, which is about 20 minutes in. Now, this isn't happening in the cytosol. The tRNA synthase, this happens in the nucleus. However, the translation occurs in the cytosol. So I'm just showing you that tRNA, the reason why we had tRNA and tRNA synthase gets charged by an ATP molecule, which connects to an amino acid. This charge is very, very, very important, because if it's not charged, you can't really bind the tRNA to our RNA to code our primary RNA transcript. So yeah, this allows for charging, and it's very, very, very important. So you need to know that you charge tRNA synthase with ATP via amino acid sequences. Now, this allows for gene expression to occur, so we need it charged. So this, which we will get to starting. Okay, cool. Okay. Nucleus. And translation occurs in the cytosol. The tRNA is where the anticodon loop is, and now we can get into the cytosol and take this one step further to get this gene, to get this expressed. So we'll come back when we need to start talking about elongation with tRNA, but for now let's go back to RNA and how the primary RNA transcript gets bound to a ribosome. Okay, so back to mRNA. Boom. Now I'm going to switch my colors to purple again. I'm going to make sure what color was my cap, my 5' cap. Was it red? What color was it? Oh. And my poly A tail was blue. Remember the PABP signal sequence is always on. Okay, there we go. And now we have another protein, an important protein, EIF4F. F4F. This binds to the 5' cap. And then we also have EIF4G. This becomes very apparent when we need to talk more about wasting diseases or polio. So we'll get back to 4G, EIF4G. EIF3 is important because it connects to EIF4F and EIF4G and binds the small subunit of the ribosome, the 40S subunit. Now this is a small diagram, should help you understand these proteins into play. But later we're going to get back into we'll go back to over the type of diseases and where they occur on these proteins and what happens to these proteins when these diseases are prevalent. So we'll get back to this. Remember where we charged our tRNA synthase earlier, and we said we come back to it here? This is where tRNA becomes important. RNAs goes from the A site to the P site to the T site, sometimes, and then it goes back to the A site, back to the P site. How does that happen? How does that work? You can't just say it. You got your 40S subunit, got your T site. It starts right here. Now, big idea. Remember, big picture is P site, A site, T site, but we're trying to get a little bit more detail. So we have our AUG, our start codon, and then UAC. So we get an amino acid, right? So that's methionine up there. Now, so the next thing happens. Got our that's our large subunit, right? 60S. How does this come into play? This comes by a protein called EIF2B. This is what brings this large subunit in. It doesn't just spontaneously come into play. You don't have the large subunit just sitting there. It comes by this protein. This is an important step. By GTP, this allows this protein to come through. Because remember, you can't just have things come through without energy. Without energy, it's not working. So EIF2B has come through GTP. Now you have that amino acid on the big head, okay? Now, what's next? What we have to do is go back we go A, then P, we go P, then A, then go back to P. So how do we do this? Another important protein, the EF2/1 protein. Don't get this confused with E2F. E2F is a transcription factor protein. EF2 is a translation factor protein. Now, EF2 pushes the ribosome forward, while EF1 takes the tRNA to the A site. So basically, pushing the ribosome forward, the tRNA isn't moving forward with it. The ribosome's moving forward, and the tRNA will go to the A site to continue to create these amino acids, but it has to still leave. So tRNA is still taken out of the equation through EF2 and 1, but the most important thing is what these two do. Now, finally, we got our ribosome. We go back to our site, and here is where EERF is another protein that bounds. Now, this finds the stop codon. This protein finds its stop codon here. Now, when it finds its stop codon, its translation stops completely, and you have your final amino acid peptide sequence, basically. Now, what's going on again? What's going on here? So you go back, and you find out that you're going from the T to the E site. What's going on? How do you get to the E site? You go back to EF2, not E2F, EF2. Remember, it pushes the ribosome forward. When it pushes the ribosome forward, this allows what to happen? To go into the T site, and allows it to stop. Now, you're finished. No, you're not finished, because gene expression is hard enough. Now we have to understand what's going to happen to the signal peptide sequence. So we have the signal peptide sequence, and basically what's happening is in the rough ER, remember, the peptide sequence gets translocated into the lumen. How does it get translocated into the lumen? Translocated into the lumen by binding SRP protein. Now, its SRP is encoded by this thing called ALU. And what ALU does is it puts SRP on the large subunit, And then finally, the subunit, the large subunit that has the SRP can bind to the subunit, the SRP receptor in the ref ER membrane. So it's your SRP receptor, and from there, binds to its SRP receptor. And remember, there's a thing called a translocon in the ref ER membrane. When the SRP receptor gets bound to the SRP on the big subunit, it opens. And this opening sequence, basically, and then after it takes your amino acid sequence, it goes through the translocon that's open and into the luminopeptide sequence. And this can have the NH3 on it, which is always at that methionine end, and it can fold and do its protein stuff, right? So we're not done still. There's still some stuff we have to go over. One important thing is, how does this get cleaved? Like, how does the amino acid just go straight into the ref ER membrane? Well, it uses a protein called signal peptidase. And that allows the cleavage to happen and allows the protein to go through. Get your NH3 plus on, and then now you could start your protein process, right? But this is just the basics, right? So we want to know what else can go wrong in this entire process, running from transcription to translation, mainly translation. But however, this process can get messed up. So what bad things can happen? So you could have a lot of things happen, actually. Let's draw a little skull for the sake of the video. It's been a long process. We're almost done, though. Skull with crossbones, right? It's got the little bones over its head. Pirates of the Caribbean. This is such a bad skull. Okay. Later we'll go over some of these proteins that can get defected, right? So let's start with let's start at the EIF4G, right? So at EIF4FG, you can have a wasting disease, which will, inhibit its process, or you could have polio. And potentially, polio can cause the EIF4G not to bind to the PABP and the EIF4G and the EIF43, and this disrupts our expression of our gene as well. This specifically is pretty fatal to the cell. I mean, it can go into the IRS channel and then cause apoptosis. Now, if you're wondering what the IRES does, basically what it does is it's like a caspase activator, and what this caspase activator does, it can cause disruption of the membrane, the gene expression. The EIF3 causes cancer. It's a cancer causing protein, so a disruption of that protein can cause that. Another thing, ricin, for all you Breaking Bad fans out there. Yeah, so ricin removes adenine from the base pairs at base pairs A4, 3, 4, 2, 4, and this disrupts EIF2B. It can become vanishing white matter. So another defect is vanishing white matter, and also another one is reticulocyte apoptosis. So that's another thing that something wrong with EIF2B could go wrong. And then another one, I think I spoke about it earlier, was diphtheria toxin. So diphtheria toxin here can also prevent elongation, and you can just stop everything if you get a defect in the EF2 protein, which pushes the ribosome forward. So a lot of these proteins and things that are going on in the translation processes, they're all very important. But the reasons why is because these diseases can get accumulated into these proteins, and if this happens, your cells can die under apoptosis and cancer as well. So don't forget E2F with the retinoblastoma protein acts as a repressor gene. So E2F by itself is an enhancer. Now, I really want to go over this last thing, with you guys, and that's basically the regulation of mRNA. And the regulation of mRNA can be caused by a few things. What are those things? Let me finish writing this. I speak way faster than I write. I don't know why I'm doing this super fast. Now, first, it could be interference with microRNA. That's one of the regulations of mRNA. Another one could be a kinase without iron. This has to do with ferritin and transferrin mRNA. We'll get back to that one. And you could also get inhibited by pharmaceuticals, such as antibiotics or things that could break the plasma, which could break some things in the membrane. And you can also get inhibited by bacteria, so, viruses, and not viruses, but bacteria itself. HUR binds can also regulate in the mRNA. AUR1 that binds can also regulate mRNA, and we'll get back to those. I just want to go a little bit deeper into akinase without iron. Now, iron regulatory protein is made from a kinase without iron. This binds hairpin loops in the mRNA. And these hairpin loops, what they do is they don't allow the translation of mRNA to happen. So if you don't have translation, you don't have a cell. You don't have a gene, right? So you don't even have a protein. You don't have a peptide, no nothing. So no translation of ferritin mRNA. However, if you have, you can have translation of transferrin without iron. So the big idea is that without iron, a kinase, you can't have it with ferritin mRNA. But with translation you can have translation with transferrin mRNA without iron. The vice versa happens when iron is bound to transferrin. So if transferrin did have iron, you don't have translation. So basically that's it. Now HUR binds the 3' end, and you can increase the stability. Now AUR binds the AU-rich regions, and it can decrease the stability. Now what does that mean? Now let's go back to the HUR. Now, if you bind HUR, what's happening is you're looking for the K site, and the K site is basically the same as AUR regions. It's the AU-rich regions, and when you look for the K site, the AU-rich regions do what? Remember, they decrease the stability. I want to talk about a quick reason why gene expression is highly regulated. The reason why it's highly regulated is you have GTP and ATP. Now, ATP itself can charge tRNA. Remember that little charging station? tRNA can get charged through the amino acid, and it's what you need to. tRNA synthase needs this in order to do its stuff. RNA polymerase also uses ATP, and here it's hesitant to incorporate adenine base pairs. ATP, it's delay translation. So if you don't have GTP, you can delay translation. This is things that are happening if you don't have them, right? You can delay translation. Why? One of the reasons why is because you can have the F2 portion of one of the proteins can't load the 60S big subunit, and that is right there. There it is. And then the other thing is that the EF1 and 2 need GTP in order to load new tRNAs, and if you remember that, that's right there. Thank you guys. This is it, and honestly, have a good day.